Our first speaker, despite having had eye surgery only a few days ago, was adamant to be here today and to connect with you. Dr. David Andrews is the Director and Senior Scientist at the Sunnybrook Research Institute. One of his research focuses is studying precision medicine and CLL. I won't go too lengthy into the bios because they're on the slides and on our website, so let's save time for them to speak. So please join me in giving a warm welcome to Dr. David Andrews.
Thank you very much for the introduction, and thank you to all of you for coming and for the opportunity to talk with you today. I'm going to talk to you about the cancer research, particularly in leukemia, that we're doing at Sunnybrook, and particularly in my own lab. This has been something that...
Microphone?
Oh, do you need the microphone? Usually I'm too loud. So I can't see that way.
But it's also for
All right.
the internet.
Take it off.
Yeah, I can
Follow the slides.
I can do. I can't point. This is fine. I've been working in this area now for more than 25 years. And one of the great success stories, you'll see a little bit about that drug here, is actually probably the main reason that AbbVie is funding this, is that they've had real blockbuster come through for CLL, and there are more in the pipe. And other companies now are following suit. So we're going to talk a little bit about those new drugs and how best to find the right patient for that drug or the right drug for that patient. So I need to tell you a little bit about chemotherapy and about cancer. So we have here two cells. We have a nice happy normal cell, and we have our cancer cell. And cancer cells are not in good condition. They have all kinds of mistakes in them. And all of us make trillions of cells, and we throw away lots and lots of cells. So the default pathway in all of our cells is to just kill themselves, because we can make more. And I'm taped to the floor. That's actually what happens all the time. And so we all have these cells that become transformed, and because of the signals that have gone wrong in these cells, then they just have death signals and they throw themselves on the sword and kill themselves. And so a tumor doesn't develop. And this is happening on a regular basis in everyone. For a tumor to develop, there has to be a block in that pathway. Okay, so once you have a block in that pathway, then these death signals are not enough to push that cell through that block. So now comes chemotherapy, and what we need to achieve with chemotherapy, and the goal here is to push that cell through that block onto the sword. Right? The problem is that when you do that, you push these guys right to the edge. Right? And that's actually what limits the amount of chemotherapy that you can give to a person, is how much you can push the normal cells and push the other cells through that block. Okay, so we would really like to improve this, and for the last 50 years, chemotherapy has been getting better and better at pushing things through the block, and it's a bit more selective, but it's the same model. The new model is, what if we could take away the block? Right? If we could take away that block altogether, then when we had the death signals, either inherent or with a very small amount of chemotherapy, we could push that cell off the wall, right? Whereas the normal cells wouldn't have to have nearly as much damage. So the idea then is to get rid of that block. And that's been the new finding, I guess, that got me into the field about 30 years ago. Our first paper on this would be 1993. So this is to give you an idea of how long it takes to actually get there. And the drug that was gotten there is a drug that inhibits this protein here, BCL2, and it's called venetoclax. And the development cost on that to phase 3 was $1 billion. Wow. So this is a long affair and very expensive. And the problem is that there are more blocks, right? So BCL2, AbbVie has a drug called venetoclax. There are drugs coming down the pipe from AstraZeneca and Serono also. BCLXL, there's a drug from the WEHI that now is owned by AbbVie. Also one's coming from some of the other companies. The AbbVie one is in late stage clinical trials. For BCLW, there is a drug. It has overlap with the other two, but there's one coming from AstraZeneca that looks really good, and there's one from Serono on MCL1 that's in clinical trials. So these are all coming. That means that we have a real question we have to answer, which is how to use these drugs, right? And to make matters worse, we have the conventional chemotherapy drugs. So here's a list of some of those over here. I just listed 10, but now you can start to see the problem. Even with that, cancer's personal, right? And so the treatment should be too. And that means we have to choose which of the 40 possible drug combinations I just showed you is going to be the best one for any individual. So how do you do that? Currently what they do is they say, well, the one that works in clinical trials for the most people is this one, so we'll give that to everybody. And then after some months and it's not working, they say, oh, well, sorry, we'll try something else. And if that was to happen in a disease like infectious diseases, you know, if you went into the hospital and you have a bacterial infection or something and they gave you the wrong antibiotic, that would be malpractice. In cancer, it's, well, we'll have to try again. And the reason is because there isn't a good way to put the right drug to the right person. And so that's what we're working on. Not just my group, but there are quite a few others that are working in this area. And I'm going to show you what we're doing in this area. So the first thing we need is a little bit of the cancer. So we get a blood sample. If it's a solid tumor, then we get a biopsy. But most of our work is with blood cancers. So we get a blood sample. And cancers are actually very messy. They're like your worst teenage nightmare. Thankfully, he is moving out of the man cave in the basement and in with his girlfriend. So they leave all kinds of rubbish around. They leave rubbish around in your bloodstream. They leave rubbish around all over the place. And so we use that. We from the blood sample, we try and get some of this rubbish and try and get information from that. In the blood cancers, the cancer cells are there, so we can get those cells and we can make use of those cells. However, the cells that are circulating, and this is now specific to blood cancers, the cells that are circulating are different than the ones that are in the bone marrow and the spleen. And the ones in the bone marrow and the spleen are the ones that we can't kill. The ones that are out here, we're really good at killing, and I tell my oncologist colleagues all the time that we've become really good at killing the wrong cells. So what we've done in my group, and with David Spanner, who we'll be talking later, he's really a pioneer in this, is to discover that the cells that are in the bone marrow and the spleen are not genetically different than the rest. They're the same. They're just being given different instructions. So if we can take the cells from the blood and give them the instructions that they're in the spleen or bone marrow, they'll behave like those cells, and we can learn how to kill those cells. And that's what we're doing, and that seems to work. So what we have to do is identify the right drug combination for any individual patient from a sample of their tumor. To do that, as I said, there's lots of detritus in the bloodstream, and we try to get DNA and RNA sequence from that, from the little pieces, also from the cancer cells themselves. We take the cells and we do this thing that I told you to make them behave like they're in the spleen or the bone marrow. That's called conditional reprogramming. And we do that in these multi-well dishes. So each one of these little wells has some cells in it, and we can put a different drug combination in each one of those little wells. Right now, for every CLL patient that comes into Sunnybrook, we test 700 different drug combinations. This little plate is about this big, and it has 1,536 little wells in it. And so we can do all of those experiments, so 700 drug combinations and controls in duplicate. And then this, in case you don't immediately recognize it from high school, that's a microscope. That's what microscopes look like these days. This one has 4 cameras and 5 lasers and takes 500,000 images a day without you being there. It has an incubator on the end. It pulls the cells, these plates out of the incubator, puts them inside itself, finds the cells, takes the pictures, puts it back, and can redo that as many times as we want, and it loads lots and lots of data. We're currently running, we just cracked 1,000 terabytes in my lab. So what we need to do is take the patient sample, put it in the plate, screen it, and then take its picture. And I just told you 900 or 1,000, we just cracked 1,000. 1,000 terabytes is a lot of data. So we need, here's the plate. So each one of these little wells is one of these experiments. And then across the top, we have all the different drugs that David or Brian or one of the clinicians I work with would prescribe. And then we have 2 different media for conditional reprogramming, so we do everything in duplicate. And over here we have all the controls. So these are control drugs that we know what they do to the cells. So some of these will cause the cells to starve. Some of them will cause them to hunker down and wait. Some cause them to die off that apoptosis cliff. Some get them just really stressed. And there's another one, it's called PDB. I ordinarily wouldn't mention it, but we're going to come back to it because it turns out to be very interesting. It changes the behavior of the cells in a way that has turned out to be very interesting. So we have all of those here so that the computer can learn what your cells look like when they're starving or dying or doing this PDB thing or whatever. So the computer uses these to understand how your cells actually function with these drugs and then returns to us that information. Okay. And then, as I said, in here, we then have the combinations and have dose response curves, so we can have different concentrations to know also the concentration that works. Okay, that's what it looks like with one picture in each of the wells. So there's a picture for each of the wells there. Typically we have 6 pictures, so 4 pictures per well. So if I blow one of those up, it looks like this. And each one of these objects that you see there is a cell. And they're stained with the different dyes. The red tells us the energy level of the cell. The blue is the nucleus, where all the DNA and RNA is. And the green is a dye that I invented that actually tells us about the physiology of what the cell is actually doing. Okay, so we have these images. So there's typically we analyze about 6,000 cells for each one of the 700 different drug combinations. If somebody would like to sit and look at these, I have a job for you. No? So we have to have a different way of doing it. So here's that image again, 6,000 images. So the computer then finds the nuclei using that blue color I just showed you, which is here. And then it draws a line around each of the cells. So it can find the cells. You can see how well it does that. It's pretty good. Finds all the cells. And then I'm going to just show you one simple measurement. We make about 100 measurements for each cell. But one measurement might be, how big is the nucleus? Because if the nucleus is too small, the cell's died. So we ask it to show us all the ones where the nucleus is too small. And it goes like that. So all the ones that are green are dead, and all the ones with the red outline are alive. Right? So we have 100 different of those measurements that the computer makes for each one of these images, and then we collate all that data in the computer. Okay? So we're back here, and as I said, 6,000 images per patient, so we need AI, right? So this is all done using artificial intelligence algorithms and machine learning that we write actually in my group. And so the goal then is to end up with data like this. And this is just a little bit of the data. I'm going to show you, and I'm going to teach you actually how to interpret it. You're going to be the computer. And on here, we have each one of these rows. There's a number here. Those numbers are the different patients. Okay? So each patient, their data goes this way across. Blue is no response, and the closer to red you are, the bigger the response is. Okay, now, over here, DMSO, that's just what we use to dissolve the drugs. So this is what the patient looks like, each of the patients look like without having any treatment, right? And each row is a patient. So the first thing that you learn from this, which should be obvious, but is not obvious to the medical system, is that everybody's different, right? Every response there is quite different. Now remember I told you about that PDB phenotype, which is it changes the cell's behavior. They actually sort of look at each other and they get little wispy tails that the computer can see. It's very hard for us to see. So we asked, have we actually taught the computer to find those? So we take all of these patient samples and we actually put some of that PDB in, so they'll all look a bit like that, right? And when we do that, that's this one here, the computer says these all are PDB, right? So it's pretty good at figuring that out, that that's what's happened. Okay? But that's when it's been trained on. What we want to know is what's happening for all the different drugs. Right? So this is just one of the drugs. Remember, there's 700 of these. So we have 700 of these panels that we have to look at. And you can see here that there are some things going on that are more reproducible. So let me show you what those are. So here's the first one. So all of these patients here, you can see when you add this drug, chlorambucil, which is frontline for CLL, all of these patients, their cells are all in these last two columns. The very last column is that cliff of apoptosis. So all of these patients are responding well to chlorambucil. The ones that have some in the second column, that's stress. So these patient cells have responded either by being really stressed out or dying. And the ones that are really stressed out are going to move to the other column if we just give them a bit more time. So all of those patients are going to respond to chlorambucil. So that's good. Then we have this group here where the response is more mixed. You can see some of them are in the last column and some of them not. And then there's these patients where these patients are not going to benefit from chlorambucil. So we should not give these patients frontline therapy. So they're not going to respond. But there's something very peculiar that we noticed when we were looking at this data. And I'm going to show it to you now because this was a discovery that we're now following up on. Look at, here's this PDB phenotype here, right? Look at it here. So the patients that came in that already had cells that were in that phenotype are the ones that are going to be resistant to frontline therapy. So we now have a test where we can take a blood sample from the patient and ask, are they likely to be resistant to frontline therapy? Do they need to have something else right from the beginning? And so we're now in clinical trial testing that hypothesis on 150 patients to see if that's really true. I think it probably will be. So the ultimate goal then is to give the physician back a report like this that they can use to identify what's the best drug combination for my patient. Yeah, I've shown you that, and it's a lot of data and a lot of work, and here's the good news. We take the sample from the patient, and 96 hours later, we're done.
And where it's done? Where is it done?
In my lab at Sunnybrook. Yeah. There's, at the moment,
everyone being tested now like this?
pardon me?
Is everyone being tested, previous patients too?
Everyone that comes into David Spanner's clinic gets tested.
What if you're Dr. Emery?
Well, I'm not getting Dr. Emery's patients at the moment. So talk to Dr. Spanner. At the moment, we're getting Dr. Spanner's patients, Dr. Lieber's patients, and we're about to start taking in AML as well. I'm waiting for I have to have a special freezer for the samples from AML, so we're waiting for that to arrive. So yeah, so that's the ultimate goal, and we have 150 patient samples in the freezer, and we're actually at the moment pulling the patient records and doing this comparison to see if the ones that we would predict were resistant to chlorambucil, that turned out to be correct. And if what we predict they're sensitive to also turned out to be correct. So we should know soon.
Are you testing for ibrutinib resistance too?
We are, yes. And I'm actually testing with a small company called ImView, a new alternative to ibrutinib that may actually be better than ibrutinib. So that's the ideas, and that's what we're doing to invent the future of healthcare.
Excellent.
And I'm happy to take questions.